Hello there, and welcome to the Sleepy Nature channel. Tonight, we are going to journey into one of the most quietly magical worlds you might never have heard of. The world of plants that grow where nothing else dares to. Imagine a hillside stained with strange colors, where the soil is full of metals that would make most plants wither and fade. And yet, here, certain small and humble plants are not just surviving. They are drinking those metals in, holding them close, and glowing with colors no ordinary leaf has ever known. Come and rest. Breathe slowly. Let the outside world go quiet and let yourself drift into this soft, strange, beautiful place. Find a comfortable position right now. Let your hands be still. Let your breath become slow and even, like the rhythm of a quiet forest after rain. There is nowhere you need to be tonight, nothing you need to do. Only this, a soft voice, a gentle story, and the slow unfolding of one of nature's most tender secrets. The world is full of places where the ground itself is unusual. Deep in certain valleys and across ancient hillsides, the soil carries something most plants cannot bear. High concentrations of metals like nickel, zinc, and cobalt. These soils were shaped over millions of years by slowly weathering rocks, by ancient seabeds lifted above the surface, by volcanic forces that pushed strange minerals up from far below. To most roots, this ground is unwelcoming. Seeds that drift in from nearby meadows often fail to sprout. Ordinary grasses push their first pale threads into the earth and then quietly stop. But there are plants that came here anyway. Long ago, so long ago that no human was watching, certain seedlings found themselves on this metallic ground and did not give up. Generation after generation, something shifted in them. They learned, in the slow and wordless way of plants, to not just tolerate the metals around their roots, but to welcome them, to pull them in, to fill their leaves and stems with substances that would have poisoned anything else. Tonight, you will meet some of these quiet wonders. You will travel gently through the places where they grow. The Serpentine barrens of California, the nickel-rich hills of New Caledonia, the zinc-dusted meadows of Belgium and Germany. You will hear about their colors, their textures, their almost unbelievable inner chemistry. And all the while, you can let your eyes grow heavy, your breath deepen, your whole body settle into rest. There is a plant called Nocaea caerulescens, and if you say it slowly, it almost sounds like a lullaby. Nocaea caerulescens. It is a small thing, modest and delicate, with tiny white flowers that would not catch your eye in a meadow. But inside its leaves, something extraordinary is happening. This plant is one of the most studied hyperaccumulators in the world. Hyperaccumulators. That is the name scientists have given to plants that absorb metals at rates hundreds or even thousands of times higher than any normal plant would. Nocaea caerulescens can pull zinc and cadmium from the soil in quantities that would make a chemist stare in disbelief. Imagine standing in a quiet Belgian meadow on a gray autumn morning. The air is cool and carries the faint smell of damp earth. The grass around you is soft and wet. And there, growing in a small cluster near a rocky outcrop, are these little plants, barely reaching your knee, their leaves a gentle gray-green. You would not know, just by looking, that each leaf is quietly working like a tiny factory, drawing metal ions up through fine root hairs, passing them through layer after layer of specialized cells, and finally storing them in a part of the leaf where they cannot do harm. The way plants protect themselves from the metals they accumulate is itself a kind of quiet miracle. Inside certain leaf cells, there are tiny compartments called vacuoles. Think of them as small, sealed rooms within the cell. The plant pumps the metal into these rooms and locks it away, separating it from the delicate machinery of the cell that must keep working perfectly. It is a bit like finding a way to store something dangerous in a safe place inside your own home. Not getting rid of it, but managing it with great care and precision. In some hyperaccumulators, the metals stored inside the leaves actually change the color of the plant. Zinc can give leaves a faint blue-gray cast. Some plants that absorb large amounts of copper develop unusual greenish-blue tints. And then there is the most astonishing case of all. A tree from the tropical forests of New Caledonia called Pycnandra acuminata. When you cut into its bark, the sap that oozes out is not white or clear or pale yellow the way sap usually is. It is blue-green, a rich, deep, unmistakable blue-green. And that color comes from nickel, up to 25% nickel by dry weight in the latex of this remarkable tree. 25%. Let that settle gently into your mind as you breathe. In the sap of a living tree, growing quietly in a warm and humid forest, there is nickel at a concentration that puts it in the same league as a metal ore. Miners search the earth for rocks that contain just a few percent nickel and call it a rich deposit. This tree holds far more than that, not in stone, but in its own living tissue, in the soft and flowing liquid that moves through its body the way blood moves through yours. Scientists first began paying close attention to hyperaccumulating plants in the latter half of the 20th century. For a long time, they were treated mostly as botanical curiosities, strange outliers in the plant kingdom, interesting but not obviously useful. Researchers would note them, measure the metal content of their tissues with careful instruments, and file away their findings. But slowly, as the decades passed, the picture began to change. It started to become clear that these plants were not just unusual, they were potentially very useful. The world produces an enormous amount of metal contamination. Old mine sites where the soil is thick with heavy metals, industrial zones where decades of activity have left the ground full of substances that ordinary plants cannot touch. Cleaning up these places using conventional methods is enormously expensive. You have to dig up the soil, transport it somewhere, treat it with chemicals, and hope for the best. But what if you could simply plant something? What if you could let a living thing do the slow, quiet work of cleaning the earth, one root at a time? This idea, using plants to pull contaminants from the soil, has a name. It is called phytoremediation. And while it is a long word for a scientific concept, at its heart, it is one of the most gentle and hopeful ideas in modern environmental science. You plant. You wait. The plant grows, pulling metals from the soil into its leaves and stems. Then you harvest the plant, taking the metals with it. Season after season, the ground becomes a little cleaner, a little safer, a little more like the soil that ordinary life can return to. You might enjoy leaving a comment below this video. Tell us if you have ever heard of these remarkable plants before, or what part of the natural world you find most quietly astonishing. Your thoughts are always a gentle pleasure to read. Let your breath go slow now. Let your shoulders drop. The story is just beginning, and there is no hurry. Outside, wherever you are, the night is settling in. The air is cooler than it was, and somewhere far away, on a hillside full of ancient metals, a small plant with white flowers is quietly doing its work, drawing the earth's hidden minerals up through its roots, holding them close, turning them into something the world has never quite seen before. There is a peace in that, a deep, slow, patient peace, and it is all around you now, as soft as the sound of wind moving through grass, as quiet as the space between one breath and the next. The Serpentine barrens of California are places that look, at first, like something has gone wrong. You drive through green hills, and then suddenly the land opens up into something pale and sparse. Low plants scattered across reddish-brown soil, outcroppings of strange colored rock, a quiet that feels different from the quiet of an ordinary field. Serpentine soil gets its name from the rock it comes from, serpentinite, a dark greenish stone that forms deep in the earth's mantle and is brought to the surface by the slow grinding of tectonic plates. It is rich in magnesium and heavy metals, low in nutrients that plants need, and it creates conditions that filter out most species ruthlessly. Only those that can truly adapt are allowed to stay. Yet, the plants that do grow on serpentine barrens are often found nowhere else on Earth. They are endemic, meaning they evolved specifically in response to these unusual soils and cannot live anywhere else. Walking through a serpentine barren in spring is a strange and tender experience. The air is dry and warm. The flowers are small but vivid, tiny blooms of yellow, white, and pale purple scattered across the sparse ground. And beneath your feet, in soil that looks inhospitable and bare, the roots of these plants are doing something that no instrument you carry can see, pulling nickel and chromium from the earth, processing it, accommodating it, building their lives around it. Breathe in slowly now. Breathe out. Feel the weight of your body against whatever is supporting you. You are safe. You are warm. And the story continues to unfold as gently as a fern uncurling in the morning light. There is a particular kind of stillness that comes when you have been walking through a wild place for a long time, and you stop finally and simply stand. Your breathing slows. Your eyes soften. The sounds around you, wind, birdsong, the distant movement of water, begin to separate from each other, and you can hear each one clearly. This is the feeling we are looking for together tonight. This is the place where the story of the metal-eating plants lives most comfortably, in that quiet, unhurried space where extraordinary things reveal themselves to those patient enough to wait and watch and simply breathe. The mountains of New Caledonia rise from the Pacific Ocean like something ancient and private. This island, roughly the size of New Jersey, sits in the southwestern Pacific, surrounded by waters of extraordinary blue. Its interior is rugged, steep ridges, narrow valleys, forests that cling to slopes so sharp that walking through them requires both patience and care. But what makes New Caledonia especially remarkable is what lies beneath those forests. The bedrock here is some of the most nickel-rich geology on Earth. Hundreds of millions of years ago, this land was part of the ocean floor, rich in minerals carried by volcanic activity from deep within the planet. When it was lifted above the sea by the slow collision of tectonic plates. It brought all those minerals with it, and they have been slowly weathering and concentrating in the soil ever since. For most life, this is a challenge. The soils here are low in calcium and phosphorus, nutrients that plants desperately need, and high in metals like nickel, cobalt, and manganese. Ordinary plants struggle. They grow pale, their leaves yellowing, their roots failing to find the chemistry they need. But New Caledonia's native flora is unlike anything else on Earth. Over 30% of the plant species found here are endemic, found nowhere else. And among them is a remarkable collection of hyperaccumulators, plants that have not just learned to survive the metal-rich soils, but have built their entire biology around them. Pycnandra acuminata grows in these forests like a quiet guardian. It is not a small plant. It is a tree, reaching heights of up to 10 meters, with broad leaves and a canopy that filters the tropical sunlight into soft, shifting patterns on the ground below. From the outside, it looks like many other trees in the forest. It has bark, it has leaves, it has roots that reach into the earth seeking water and minerals. But inside, it carries its secret, that extraordinary blue-green latex, rich with nickel at concentrations that no other plant on Earth is known to reach. Imagine walking through the New Caledonian forest on a warm afternoon. The air is humid and smells of earth and flowers. Light filters through the canopy in long, gentle rays. You stop beside this tree and make a small cut in its bark with a careful instrument, and what flows out is not the color you expect. It is blue-green, not a pale or uncertain color, but a definite, deep, beautiful blue-green, like the water of a sheltered lagoon seen from above. The tree is not in pain. It does not react. It simply goes on being what it is, a living vessel for one of nature's most astonishing chemical achievements. Scientists who study Pycnandra acuminata have spent years trying to understand exactly how it manages such high nickel concentrations without destroying itself. The nickel is bound to organic compounds within the latex, molecules called organic acids that hold the metal in a form that does not interfere with the tree's cellular functions. The tree has essentially built a chemical cage around the metal, a molecular structure that allows it to carry nickel through its tissues the way other trees carry sugars and water. It is peaceful chemistry, patient, precise, and entirely invisible to the naked eye. In the surrounding forest, other plants are doing similar things at smaller scales. Species of Renoria, a genus of small shrubs, are known to accumulate nickel in their leaves. Certain ferns push their fronds up through the metal-rich soil and quietly fill them with zinc. Each species has its own particular chemistry, its own preferred metal, its own set of molecular tools for managing what it absorbs. The forest is, in a sense, a community of quiet specialists, each one doing its particular work without disturbing the others. Let your mind rest on that image for a moment. A forest full of plants, each one quietly doing something extraordinary, none of them hurrying, none of them competing loudly, all of them simply growing, absorbing, adapting. There is a lesson in that softness, though we will not make it too sharp tonight. Just let it be present, the way a warm feeling is present in a room without needing to be named. If this kind of quiet wonder is what you look for in your evenings, you might enjoy exploring our playlist. There are many more stories just like this one, waiting for you whenever you need to slow down and breathe. The link is always there for you whenever you are ready. The story of hyperaccumulating plants is also quietly a story about time. These plants did not develop their abilities overnight. The process of evolution that led to hyperaccumulation took place over thousands or perhaps hundreds of thousands of generations. Each generation faced the same challenge: metallic soils, competition from other species that could grow elsewhere, pressure to survive and reproduce. And in each generation, those individual plants that happened to carry slight variations in their chemistry, a protein that bound nickel a little more efficiently, a transport molecule that moved zinc more effectively across cell membranes. Those plants survived a little better, reproduced a little more successfully, passed their variations on. This is evolution working at its most patient and most beautiful. Not dramatic transformation, not sudden leaps, but the slow accumulation of small advantages over immense stretches of time. The plant that grows on a nickel-rich slope in New Caledonia today is the living result of all that patience. It carries in its cells the accumulated chemistry of countless generations of ancestors who faced the same metallic soil and found slowly a way not just to endure it, but to make it their own. The roots of a hyperaccumulator are worth thinking about on their own. In the soil, roots are surrounded by a complex world: bacteria, fungi, other roots, particles of mineral and organic matter, water moving through tiny pores, chemical gradients shifting with temperature and rainfall. In this world, a hyperaccumulator root is doing something actively unusual. It is releasing compounds into the soil, acids that help dissolve metal ions, chemicals that encourage certain bacteria to make metals more available. The root is not passive. It is in dialogue with the soil, changing it slightly, making it yield what the plant needs. In some cases, the soil around hyperaccumulator roots is actually different from the soil just a few centimeters away. The chemistry shifts. Certain metals are more dissolved, more mobile, more available to the hungry root hairs that spread through the earth in thin, white threads. And as the plant takes up those metals, the soil changes again, slowly, incrementally, season by season. The plant and the soil are shaping each other in the way that all living things and their environments shape each other over time. Breathe slowly. Let that image drift through your mind like a leaf on still water. Roots reaching into earth, drawing up metals, leaves filling slowly with something that shines. The night is quiet around you. Your breathing is steady and soft. There is nothing to solve tonight, nothing to figure out. Only this gentle story, moving forward at its own quiet pace, the way rivers move, always forward, always soft, always finding the lowest and most peaceful path. The word hyperaccumulator sounds scientific and perhaps a little cold, but the thing itself is anything but. It is a plant. It grows in sunlight. It flowers in spring. It feeds insects and shelters small creatures. It is part of a community, embedded in a landscape, rooted in place. The fact that it also carries metals in its tissues is just one part of what it is, correct? An extraordinary part, yes, but woven into a life that is, in most ways, as ordinary and beautiful as any plant life you might find anywhere on Earth. A flower opening in the morning light, a root drinking in the cool darkness of the soil, a leaf turning slightly to catch the sun. These things happen in the metal-rich barrens just as they happen in any meadow or garden, with the same quiet rhythm, the same unhurried grace. There are currently around 700 known hyperaccumulating plant species on Earth. 700. That number sounds large, but consider that there are estimated to be over 400,000 plant species in total. So, hyperaccumulators represent less than 0.2% of all plants. They are rare. They are specialized. They are tucked into corners of the world that most of us will never visit. Serpentine outcroppings, old mine tailings, ultramafic soils hidden in tropical forests. But they are there, patient and still and quietly remarkable, growing at the edges of the ordinary world, their leaves full of something unexpected and strange. And somewhere tonight, right now, while you rest and breathe, one of them is out there in the dark, drinking from the earth, doing what it was always meant to do. Let your eyes be heavy now. Let your hands be soft. The second part of our story is complete, and the rest waits for you gently, like the next page of a book you have been reading by firelight. There, whenever you are ready, unhurried, warm. It is also worth pausing to think about how scientists actually discover that a plant is a hyperaccumulator. You cannot tell just by looking. The color might offer a hint in the most extreme cases. That blue-green sap of Pycnandra, the slightly unusual tint of certain zinc-accumulating leaves. But for most hyperaccumulators, there is no visible sign. The leaves look ordinary. The flowers are modest. To know what is truly inside, you have to measure. Researchers collect leaf samples, dry them carefully, then use analytical instruments, mass spectrometers, atomic absorption spectrophotometers, to measure the precise concentrations of metal atoms within. Sometimes a plant that looks completely unremarkable turns out, under analysis, to be holding nickel or cobalt or thallium at concentrations that seem almost impossible. And in that moment, the ordinary becomes extraordinary. Not because the plant changed, but because the measuring revealed what was always there. Zinc is one of the most common metals in the Earth's crust, and it is also one of the most commonly accumulated by hyperaccumulating plants. In small amounts, zinc is actually essential for plant life. It plays a role in enzyme function, in the processing of chlorophyll, in the regulation of growth. Every plant needs some zinc, but hyperaccumulators take this ordinary need and carry it to a remarkable extreme. Where a typical plant might contain a few hundred parts per million of zinc in its leaves, a zinc hyperaccumulator can contain 10,000, 20,000, or even more. The plant is not just meeting a need, it is filling itself beyond any ordinary requirement, storing zinc in its tissues at concentrations that would be toxic to any ordinary plant. Noccaya caroescens, that small, modest plant with the soft white flowers that we met in the beginning of our story, is one of the most studied zinc hyperaccumulators in the world. Researchers have taken it apart, studied it at every level, from the behavior of its roots in the soil all the way down to the individual protein molecules that carry zinc ions across cell membranes. They have found that this plant has an unusually high number of certain transporter proteins, molecular machines that sit in cell membranes and actively pump zinc from one side to the other. In an ordinary plant, these transporters work at normal capacity. In Noccaya caroescens, they are working continuously, moving zinc with a diligence and efficiency that has no parallel in the plant kingdom. Picture those transporters now, not as mechanical things, but as living molecules, proteins folded into precise shapes, sitting in the thin membrane of a cell. Each time a zinc ion drifts close, the transporter catches it, changes shape slightly, and pulls it through the membrane to the other side. Then it resets, ready to catch another. Millions of times a day, in millions of cells across every leaf of every plant in the colony, this quiet work continues. No sound, no effort, no awareness, just the endless, patient movement of metal through living tissue, as regular and calm as breathing. The zinc that accumulates in these leaves has an interesting effect on the insects that might otherwise eat them. Many hyperaccumulators are remarkably resistant to herbivory, the plant-eating activity of caterpillars, beetles, aphids, and other creatures that make their living by consuming plant tissue. Scientists have found that caterpillars placed on the leaves of zinc-rich Nokia caerulescens often eat less, grow more slowly, and survive less well than caterpillars on low-zinc plants. The metal acts as a natural defense, a kind of armor built not from thorns or tough fibers, but from chemistry. This is one of the theories for why hyperaccumulation evolved in the first place. Among the several hypotheses that researchers have proposed, the elemental defense hypothesis is one of the most widely discussed. The idea is simple and elegant. Plants that accumulated metals in their leaves gained protection from the creatures that would otherwise eat them. In environments where metals were abundant in the soil, the plants that developed this chemical armor had an advantage. They were eaten less, survived better, reproduced more successfully. Over generations, the ability to accumulate metals was shaped and refined by the constant pressure of hungry insects and other herbivores. Of course, nothing in evolution is ever quite that simple. There are other hypotheses, too. One suggests that metals might protect against fungal pathogens, the microscopic organisms that invade plant tissue and can cause disease. Metal-rich leaves might create an environment where pathogenic fungi struggle to establish themselves. Another hypothesis proposes that metal accumulation might help the plant deal with drought or nutrient imbalances in ways that are not yet fully understood. And it is possible that in different species, different pressures were most important, that hyperaccumulation evolved not once, but many times, in many different places, for reasons that were not always identical. The thought that hyperaccumulation evolved independently in many different plant lineages is itself remarkable. Taxonomists, scientists who study the relationships between species, have found hyperaccumulators scattered across dozens of different plant families. They appear in the mustard family, in the violet family, in the daisy family, in the fern family, and in many others. These plants are not closely related to each other. They did not inherit their metal-accumulating abilities from a single common ancestor. Instead, evolution found the same solution, or something very like it, again and again, in different lineages, on different continents, in different geological settings. Scientists call this pattern convergent evolution, the independent arrival at similar solutions to similar challenges. It is one of the most compelling phenomena in all of biology because it suggests that some solutions are so effective, so well matched to their challenges, that life tends to find them regardless of where it starts. The wing, the eye, the streamlined shape of a fast swimmer, These have all evolved independently many times. And now we can add to that list the ability to drink metal from poisoned soil and turn it into a kind of living chemistry that keeps the plant safe. Do you find yourself drawn to the quiet corners of the natural world? We would love to hear about them in the comments, the unusual plants or landscapes you have discovered, the moments of unexpected wonder in your own patch of earth. Every comment adds something warm to this little community we are building here together. Your body is settling now. Perhaps your breathing has slowed a little without you noticing. Perhaps your hands have unclenched, your feet have relaxed. The small tensions of the day have begun to loosen their grip, one by one, quietly, the way the colors in a sunset soften as the light fades. This is exactly the right state for the rest of the story. There is no urgency here. The plants we are describing have been doing their work for millions of years and will continue long after any of us. There is something deeply restful in that continuity, in the knowledge that the Earth's quiet processes go on, patient and untroubled, through all the seasons and all the years. The meadows of Central Europe have their own version of this story. In parts of Belgium, Germany, and Luxembourg, there are patches of grassland growing on soils that contain elevated levels of zinc and lead, the legacy of centuries of mining activity that has now long since ceased. The mines are closed, the smelters are cold, but the metals they left behind in the soil remain, distributed through the earth in concentrations that prevent ordinary grass from establishing itself. These places are sometimes called metallophyte grasslands, grasslands of metal-loving plants, and they have developed their own quiet ecology over the decades and centuries since the mines closed. In these meadows, Nokia caronescens grows alongside other specialists, plants that have learned through their own long processes of adaptation to manage metallic soils. There are certain species of violet that can tolerate zinc concentrations that would stop most plants. There are grasses with tough, narrow leaves that persist where broader-leaved species fail. And in the spaces between, the soil itself is being slowly changed by all this living activity. Metals taken up into plant tissue, cycling back to the ground when leaves fall and decompose, moving through the food web in ways that scientists are still working to fully trace. Rest now. Let the image of those quiet European meadows be present with you. The gray-green of metal-tolerant leaves, the small white flowers of plants that ask for nothing except the freedom to grow in their own unusual way. The night is full of such places if you imagine it widely enough. Small patches of the extraordinary hidden inside the ordinary landscape of the world. And you're here, warm and still, learning their names. In the language of botany, a metallophyte is any plant adapted to grow on metallic or mineralized soils. The word combines the Greek for metal with the Greek for plant. A metal plant, a plant of metals. Metallophytes include both hyperaccumulators, plants that actively draw metals into their tissues in large quantities, and excluders, plants that have developed ways of keeping metals out, preventing them from entering their roots or moving from roots to leaves. Both strategies are valid, both have evolved, and both result in plants that can do something most of their relatives cannot: persist and even thrive in places where the chemistry of the earth would defeat anything less specialized. The excluders have their own quiet elegance. They build barriers in their roots, layers of cells with modified walls that intercept metal ions and prevent them from traveling upward. They express proteins that bind metals near the root surface, neutralizing their potential to cause damage. Some excluders even change the chemistry of the soil around their roots, raising the pH or releasing compounds that cause metals to precipitate out of solution and become unavailable for uptake. These plants are not accumulating metal, they are defending against it. And in their defense, they reveal just as much ingenuity as the plants that welcome metal in. Both strategies, accumulation and exclusion, are part of the same larger story, the story of life finding a way, of roots meeting the earth and neither giving up nor being defeated, but instead entering into a long, slow negotiation that produces, over time, something new, something that fits, something that belongs in its own particular way exactly where it is. Let the story carry you now, soft and unhurried, into a deeper kind of rest. Cobalt is a metal with a color associated with it, a deep, vivid blue that has been used by artists for centuries. Cobalt blue pigments colored the glass of medieval cathedrals, the tiles of ancient Persia, the pottery of China's Tang dynasty. There is something of the sky in that color, a depth and clarity that seems to go on forever when you look into it. And cobalt is also quietly one of the metals that certain plants have learned to accumulate. In the copper belt of Central Africa, a broad sweep of territory crossing parts of what is now the Democratic Republic of Congo and Zambia, the geology is extraordinarily rich in copper and cobalt. For thousands of years, people have mined these metals here. The landscape carries the marks of that long history, areas of disturbed earth, soils leached of their nutrients by centuries of exposure, places where the ordinary cover of vegetation has been stripped away and not returned. But in many of these places, something is growing. Small plants, tough and specialized, have colonized the metallic soils and established themselves in communities that exist nowhere else. These plants are sometimes called copper flowers, though they are not a single species, but a whole flora, many different species from different families that share the ability to grow where copper concentrations would kill most plants. Scientists who have studied these copper-tolerant floras have found that they often contain both excluders and accumulators, plants with different strategies sharing the same unusual habitat, finding their own particular ways of being at home in metal-rich earth. Among the most remarkable of these copper belt plants are certain species of Hyperaccumulators, low-growing herbs with small, modest flowers that contain copper in their tissues at concentrations that make them almost mineral by nature. Researchers have found Hyperaccumulators cuttingense growing on soils with copper concentrations so high that even specialist mining equipment would register them as notable. And the plant simply grows there, quietly, season after season, doing what it has always done. Imagine the silence of that landscape, a flat, dry plateau in the heart of Africa, the sky enormous overhead, the light at midday, white and intense. The earth is reddish-brown, the color of old metal, the color of iron and copper mixed and baked in the sun. And across this earth, in scattered patches, the copper flowers grow, small and unspectacular to the eye, extraordinary in their chemistry, their leaves full of a metal that has colored the art of civilizations and powered the electrical systems of the modern world. There is a particular kind of beauty in understatement, in things that do not announce themselves, that carry their extraordinary qualities quietly and without display. The copper flowers of the African plateau have this quality. They are not showy. They do not draw the eye with brilliant colors or dramatic forms. They simply grow, and in growing, they perform one of the most chemically demanding tasks in the plant kingdom, as naturally and effortlessly as a dandelion pushing through a crack in a pavement. The roots of these plants are very fine. Root hairs, the thinnest and most delicate extensions of a root system, are where most water and nutrient absorption happens, and in hyperaccumulators, they are where the first encounter between plant and metal occurs. These root hairs can be thinner than a human hair, extending out into the spaces between soil particles in every direction, probing the chemistry of the earth with exquisite sensitivity. When a copper ion drifts into the water film surrounding a root hair, specialized proteins on the surface of the hair recognize it and begin the process of bringing it inside. Think of that. A molecule so small it cannot be seen, drifting through the water in the darkness of the soil, finding the surface of a root hair thinner than a thread, and being recognized and drawn in. The entire transaction is invisible, silent, occurring in darkness at a scale far below anything we can directly observe. And yet, it is happening constantly across the root systems of every hyperaccumulator in every metal-rich place on Earth. Right now, as you lie still and breathe and let the night deepen around you. Phytomining is a word that sounds industrial and modern, but the idea it describes connects to these ancient, quiet plants in a profound way. Traditional mining is a violent process in some sense. It involves moving enormous amounts of earth, processing rock with water and chemicals, separating metals from the material around them at great expense of energy and resources. Phytomining imagines a different approach, planting hyperaccumulators in metal-rich soils, growing them through a season, harvesting the biomass, and then extracting the metals from the plant material. The plants have already done the difficult work of concentration. What remains is essentially a processing task. Researchers have calculated that in some cases growing and harvesting hyperaccumulators on nickel-rich ultramafic soils could produce yields of nickel that are economically competitive with conventional mining, particularly for soils where metal concentrations are too low to make traditional mining economically viable. These sub-economic ore deposits, places where there is metal in the ground, but not enough to justify the cost of digging and processing, might become productive through phytomining in ways that are gentler on the landscape and cheaper to operate. We are still in the early stages of exploring this possibility. Phytomining has been demonstrated experimentally, but it has not yet been widely deployed commercially. There are challenges. Hyperaccumulators tend to grow slowly and produce relatively little biomass. The harvest and processing of metal-rich plant material raises its own questions about efficiency and cost, and the landscapes best suited for phytomining are often remote and difficult to work in. But the principle is sound, and as research continues, the plants themselves are being studied more carefully. Their growth rates, their metal uptake efficiencies, their soil requirements. in the hope of understanding them well enough to help them do even more than they already do. If you would like to explore more quiet corners of the natural world with us, our playlist has many more stories waiting, each one a gentle journey through some part of the earth that has something surprising and peaceful to share. You are always welcome there. Your breath is slow and even. The room around you is dim and quiet. Somewhere in the earth beneath you, beneath the floors, the foundations, the layers of soil, metal is present. It is always present in the minerals that make up the ground, in the rocks from which all soil was eventually made. We live above a mineral world that is ancient and deep and full of quiet wonders. And on certain hillsides and plains and valleys, the plants we have been meeting tonight are reaching down into that mineral world and drawing it upward through their roots and stems and into their leaves, where it becomes something unexpected, a chemical story written in living tissue, readable only to those patient enough to look closely and ask the right questions. Let that thought drift gently through your mind and dissolve, like a cloud softening at the edges as the day ends. You do not need to hold it. Just let it be present for a moment, and then let it go, the way all things are gently let go when the body is ready to rest. Thallium is one of the rarer metals in the Earth's crust, and it is also one of the most toxic. Even in small amounts, thallium compounds can cause serious harm to living things. It has been used historically as a rodenticide and in darker chapters of human history as a poison. Yet, there are plants that not only tolerate thallium in their soils, but actively accumulate it. Aberus intermedia, a small flowering plant from the cabbage family with clusters of white flowers that look almost cheerful against the stark soils where it often grows, can accumulate thallium to concentrations in its leaves that are simply staggering. Researchers have measured thallium levels in this plant's leaves at concentrations thousands of times higher than in the surrounding soil. How does a plant survive with so much of one of the Earth's most toxic elements in its tissues? The answer involves the same molecular strategies we have seen before: compartmentalization, chemical binding, specialized transport, but taken to an extreme that represents one of the most dramatic examples of biological adaptation known. The plant's cells are not simply tolerating the thallium. They have evolved to manage it so precisely that its toxicity is effectively neutralized. The thallium is there in large quantities, but it is rendered harmless by the cellular architecture that surrounds it. Walking through a field where Iberis intermedia grows is an ordinary enough experience. White flowers, green leaves, the buzz of pollinators moving among the blooms. Nothing in the appearance of the plant betrays what it is carrying inside. The insects visiting its flowers to collect nectar and pollen are exposed to the thallium in the plant's tissue, and researchers have found that the concentration of thallium in the bodies of insects that feed on these plants is also elevated. The metal moves into the food web quietly and invisibly, tracing its way through the community of life that surrounds these unusual plants. The world of the hyperaccumulator is not isolated. It is connected always to everything around it. Close your eyes now, if they are not already closed. Let the breath come and go without effort. Feel the quietness of the night settling around you like the softest of blankets, and let the story of the metal-eating plants continue to drift through your mind as gently as smoke rising from a candle curling upward and then fading into the still air above. There is a small island in the southwestern Pacific called New Caledonia, and we have visited it already in this story. But there is another island group nearby, the Philippines, where the story of metal-loving plants has taken on a particularly interesting dimension in recent years. The Philippines sits on the Pacific Ring of Fire, a great arc of volcanic activity and tectonic movement that circles most of the Pacific Ocean. This geological setting has given the archipelago some of the richest mineral deposits in Asia, including large reserves of nickel-rich laterite soils in places like Palawan, Mindanao, and Surigao. And where there are nickel-rich soils, there are, as we now understand, plants that have learned to live with them. Raynoria bengalensis is a shrub found in the nickel-rich forests of the Philippines. Scientists have measured nickel concentrations in its leaves at levels that make it one of the highest nickel hyperaccumulators found in Asia. Walking through the forests where it grows, you would find it among many other plants, its leaves indistinguishable in color and texture from its neighbors. No blue-green sap, no unusual tint, no visual signal of what it carries. The extraordinary is hidden inside the ordinary, as it so often is. The discovery of hyperaccumulators in the Philippines has generated genuine scientific excitement, because these plants grow in a country where nickel mining is economically important, and where the impacts of that mining on local ecosystems are a matter of ongoing concern. The possibility of using native hyperaccumulators for phytomining or for the rehabilitation of mined lands, where soils have been stripped and metal concentrations in remaining areas are high, is being explored with real interest by researchers working with local communities and government agencies. There is something quietly hopeful about that. The idea that the plants native to a place might hold part of the answer to some of the challenges that place faces, that the knowledge built into the biology of a local shrub over thousands of years of evolution might become useful in ways that no one anticipated when the plant was first described. It is a reminder that biological diversity has value beyond what we can currently measure or imagine, that in the species we have not yet fully studied, in the plants growing unnoticed at the edges of disturbed ground, there may be answers to questions we have not yet learned to ask. The soil around a nickel hyperaccumulator in the Philippines is alive in more ways than one. Bacteria play a crucial role in the availability of nickel in the soil. Certain bacterial species break down mineral structures and release nickel ions that the plant can absorb, while others form relationships with plant roots that may help the plant deal with the metal once it has been absorbed. The community of microorganisms in the soil around these plants is often different from the community in nearby soils without hyperaccumulators. The plants are shaping their microbial environment, and the microbes are shaping the plant's experience of the metal-rich ground. This mutual shaping, this constant dialogue between plant and soil microbiome, is one of the frontiers of current research in hyperaccumulation biology. Scientists are beginning to understand that the ability to accumulate metals is not a property of the plant alone, but of the plant soil system as a whole. The bacteria in the soil are part of the story. The fungi that form partnerships with roots, mycorrhizal fungi, whose fine threads extend far into the soil and bring water and nutrients back to the plant, may also be involved in metal uptake and transport in ways that are not yet fully characterized. Picture the soil beneath a Philippine forest, dark, rich with organic matter, threaded through with the white filaments of fungal hyphae and the thinner threads of root hairs, populated by billions of bacteria doing their quiet chemical work. In this underground world, a Rhineria bengalensis root is surrounded by activity it has always known and adapted to. The exchange of molecules between root and soil, between plant and microbe, has been going on in this particular way for as long as this species has existed. It is a relationship as intimate and as ancient as any in the natural world. You are still here, breathing. The night is still around you. Perhaps the room has grown a little darker, a little quieter. Perhaps the sounds from outside have faded into the background, becoming less distinct, more like the general hum of the world going on without requiring your attention. This is exactly right. There is nothing here that needs your active mind. The story can come to you, drift through you, and leave without asking you to hold on to it. Arsenic is a word that carries a certain drama. It is the stuff of mystery novels and dark historical chapters associated with poison and secrecy and harm. But arsenic is also a naturally occurring element found in soils around the world, particularly in areas with volcanic geology or in places where certain mining activities have taken place. And there are ferns, soft, green, gracefully arching ferns, that have learned to accumulate arsenic in their fronds at concentrations that are themselves almost unbelievable. Pteris vittata, the Chinese brake fern, is probably the most famous arsenic hyperaccumulator in the world. It is not a rare or exotic plant. It grows widely across tropical and subtropical Asia, and it has naturalized in parts of the Americas and elsewhere. You might have seen it without knowing what it was, growing in a garden or along a path, its fronds long and feathery and a cheerful, ordinary green. Nothing about its appearance suggests that it is one of the most extreme metal accumulators known to science. But Pteris vittata can absorb arsenic from contaminated soils and accumulate it in its fronds at concentrations hundreds of times higher than what most plants could tolerate. The discovery of Pteris vittata's arsenic hyperaccumulating ability, reported by researchers at the University of Florida in the year 2001, was genuinely surprising to the scientific community. Ferns were not well represented among known hyperaccumulators at that time, and arsenic was not a metal that researchers expected to find accumulated at such extreme concentrations. The paper reporting the finding was widely discussed and led to a significant expansion of interest in the practical use of hyperaccumulators for remediation of arsenic-contaminated soils. Arsenic contamination is a serious problem in parts of the world, particularly in Bangladesh and West Bengal, India, where naturally occurring arsenic in groundwater has caused widespread health problems in communities that depend on well water. On a different scale, arsenic contamination of soils occurs around old mine sites, industrial facilities, and areas where arsenic-containing pesticides were used in the past. The prospect of using a fern, a plant that is easy to grow, adapted to a wide range of conditions, and capable of absorbing arsenic with remarkable efficiency, to slowly clean these soils is one that researchers have pursued with considerable energy since that initial discovery. Breathe slowly. Let your body be heavy. The fronds of the Chinese brake fern are long and soft, And if you imagine them now, they move a little in a warm breeze, arching gently, catching the light, green and calm, and quietly carrying their invisible burden with complete, untroubled ease. This is one of the most peaceful images in all of science. A beautiful, ordinary fern doing something extraordinary without any apparent effort, simply by being what it is. Since the discovery of Terras vittatus arsenic hyperaccumulation, researchers have identified other fern species with similar abilities. Phytoremediation calomelanos, the silver fern, is another example. Its fronds coated with a pale, silvery powder that gives it an almost luminous appearance in certain lights, and its tissues accumulating arsenic from contaminated soils with an efficiency that makes it a promising candidate for remediation work. The silver fern is especially common in tropical regions, which is relevant because arsenic contamination is also particularly prevalent in tropical and subtropical areas with certain geological profiles. The variety of plants that have developed hyperaccumulating abilities is one of the things that most impresses researchers who study this field. Not just herbs and shrubs, but trees. Not just flowering plants, but ferns. Not just one or two families, but dozens. This distribution suggests that the selective pressure for metal accumulation has been widespread across ecosystems, appearing in many different evolutionary lineages because the challenge it addresses, surviving in metal-rich soils, has itself been widespread. Metal-rich soils are found on every continent, and on every continent there are plants that have found their own particular way to meet that challenge, to make themselves at home in the difficult and unusual earth. Let the thought of all those different plants in all those different places doing their quiet chemical work be like a soft blanket of living things spread across the world. The Chinese brake fern in its warm valley, the copper flowers of the African plateau, Pycnandra in its Pacific forest, Nocaea in its Belgian meadow, all of them breathing and growing and accumulating, night and day, in sun and rain and wind, in a world that barely knows they are there. And now, quietly, And that knowledge is a small and beautiful thing to carry with you into sleep. There is a quality of light in the late afternoon, the hour before the sun sets, that botanists and naturalists often speak of with particular affection. The light is low and warm, coming from an angle that makes every surface catch it differently. Leaves glow, shadows are long and soft. The air seems to hold the day's warmth without urgency, offering it gently before the night comes. In this light, even an ordinary meadow becomes something worth pausing for. And in the fields where hyperaccumulators grow, the metallophyte grasslands, the serpentine barrens, the disturbed margins of old mining sites, this late light finds the small flowers and modest leaves of remarkable plants and makes them beautiful in the way that all ordinary things become beautiful when the light is right and you are still enough to notice. The science of growing things in metal-rich conditions is called metallophyte ecology, and it is a field full of patient people, researchers who spend seasons in unusual places collecting soil samples and plant specimens, measuring metal concentrations with careful instruments, mapping the distribution of species across landscapes that most visitors would pass through without noticing anything unusual. These scientists often have a particular quality about them, a kind of deep patience, a capacity for attention to slow and quiet processes a genuine affection for the organisms they study. It takes a certain kind of curiosity to find beauty in a plant that most people would overlook, growing in a landscape that most people would consider barren. One of the pioneers of hyperaccumulator research was a botanist named Reginald Brooks, who spent decades studying plants on ultramafic soils in Australia, New Caledonia, and elsewhere. Brooks was instrumental in developing the formal definition of hyperaccumulation, the threshold concentrations that distinguish an ordinary plant from a hyperaccumulator, and he worked tirelessly to document the diversity of species that met that threshold. His research, carried out over a long career, laid much of the foundation on which current understanding of hyperaccumulating plants is built. He was, by all accounts, someone who genuinely loved what he studied, who found in these quiet, unusual plants a kind of wonder that sustained a lifetime of work. There is a specific threshold that defines a hyperaccumulator for zinc. A plant must accumulate at least 10,000 parts per million of zinc in its leaves to qualify. 10,000 parts per million is 1% by weight, meaning that 1 out of every 100 grams of leaf tissue is zinc. For a metal that is present in ordinary plant tissue at concentrations 100 times lower than this, that is an extraordinary departure from the norm. It is the difference between carrying a backpack and carrying a backpack filled entirely with metal weights. The structure of the plant manages this load through chemistry rather than physical reinforcement, binding the zinc in ways that neutralize its chemical activity and keep the cellular machinery of the leaf working normally, despite the presence of so much of what should, by ordinary rules, be toxic. The leaves of high-zinc Nocaia caerulescens have been analyzed in remarkable detail, using techniques that allow researchers to map the distribution of elements within a tissue at the scale of individual cells. Scientists have shown exactly where the zinc goes in a hyperaccumulator leaf. It concentrates in the vacuoles, those small storage compartments inside the cells, and particularly in the cells of the epidermis, the outermost layer of the leaf. The mesophyll cells in the interior of the leaf, where photosynthesis takes place, contain less zinc. The plant appears to keep the photosynthetically active tissue as protected as possible, concentrating the metal in the outer cells where it is less likely to interfere with the light-capturing chemistry that powers the plant's life. This spatial organization within the leaf is one of the things that impresses scientists most about hyperaccumulators. The distribution of metals is not random. It is patterned, organized, controlled by the same cellular machinery that controls every other aspect of the plant's internal chemistry. The plant knows, in the wordless way that all biology knows things, exactly where to put what it has accumulated and how to keep it there without causing harm. It is a masterwork of cellular architecture accomplished without consciousness or intention, shaped entirely by millions of years of evolutionary refinement. The word vacuole comes from the Latin for empty, but vacuoles are anything but empty. They are the storage organs of the plant cell, filling with water to give the cell its turgor, holding pigments, organic acids, sugars, and, in hyperaccumulators, metals. The central vacuole of a plant cell can occupy most of the cell's volume, like a large water balloon surrounded by a thin skin of cytoplasm and all the organelles that carry out the cell's metabolic work. In a hyperaccumulator, this central vacuole is where much of the accumulated metal ends up, sequestered safely away from the sensitive chemistry of the rest of the cell. Think of the cell as a small room, and the vacuole as a sealed cabinet in the corner of that room where things are stored. The rest of the room goes on with its ordinary life, cooking, working, sleeping, while the cabinet holds its contents quietly and safely. The cell photosynthesizes, respires, divides, and grows, all the while keeping its stored metal separate and managed. This is not a compromise or a limitation. It is a beautifully engineered solution, arrived at over countless generations of trial and error in the long university of evolution. We are so glad you are spending this evening with us, resting and listening. If you are enjoying this gentle journey, please feel free to like this video. It helps others find their way here when they need a quiet corner of the natural world to rest in. Your support makes it possible for us to keep sharing these stories. The history of mining and human civilization is long and complex. Metals have been extracted from the earth for thousands of years, shaping the development of technology, trade, art, and warfare. The tools that built ancient temples, the coins that moved through medieval markets, the rails of the Industrial Revolution, the wires of the modern electrical grid, all required metals taken from the ground, refined and shaped and used in the service of human purposes. This extraction has left its marks on landscapes around the world. Mine tailings, the waste rock and processed material left over after metal extraction, cover large areas in many countries. Acid mine drainage can make streams and rivers inhospitable to ordinary life. In some regions, the effects of past mining activity persist for centuries after the mines themselves have closed. It is in this context that phytoremediation and phytomining take on their full significance, not as small laboratory curiosities, but as potentially practical tools for addressing some of the consequences of humanity's long relationship with the minerals of the earth. The plants we have been discussing, these modest, quiet, extraordinary organisms, may have a role to play in helping to repair some of what has been disrupted. Not quickly, not completely, but slowly, patiently, season by season, in the way that all healing in the natural world proceeds, one small step at a time, unhurried, persistent, and ultimately effective. Let your breathing be easy now. Let the night hold you. The earth is always in the process of healing itself. Through the slow weathering of rock into soil, through the gradual colonization of disturbed places by whatever life can make a foothold there, through the patient chemistry of plants and bacteria and fungi working together to build something stable and living from what was damaged or bare. This process is always happening, too slow for most of us to see in a single lifetime, but continuous and real. The hyperaccumulators are part of it. They are among the first responders to metallic disturbance, the pioneers that arrive when other life cannot, that begin the slow work of changing the chemistry of a place into something that more life can eventually use. In their patience, they are teaching us something about slowness, about the virtue of being adapted to where you actually are, rather than requiring the world to be different from what it is, about finding a way to live with difficulty rather than being defeated by it. These are not lessons that need to be stated directly. They are present in the simple fact of what these plants are and what they do. And on a quiet evening, with your body at rest and your mind growing soft at the edges, They can be received in the way that the best lessons are received, not as instructions, but as a feeling, a sense of something being right, a quiet acknowledgement that the world, in its most unexpected corners, knows exactly what it is doing. The soil of an old mine site is not a simple thing. It is a complex mixture of disturbed geology, rock that has been crushed and moved and processed, stripped of some of its minerals and left enriched in others. It has been exposed to air and water and chemical treatment in ways that natural soil never has. Its structure is often poor, compacted or loose, lacking the organic matter and biological life that give healthy soil its character and its capacity to support plant life. Reclaiming these sites, bringing them back to something that supports living communities, is one of the significant environmental challenges of our time. Hyperaccumulators are not the whole answer to this challenge, but they are part of it. They can establish themselves in conditions that defeat other plants. They can begin to add organic matter to the soil as their leaves fall and decompose. They can create microclimates and soil conditions that allow other, less metal-tolerant species to gradually establish themselves. They can reduce the mobility of metals in the soil, making them less likely to leach into groundwater. Over time, over many decades, a landscape colonized by hyperaccumulators and their associated species can develop towards something more complex and stable. Not quite what was there before the mining began, perhaps, but a living place again, with its own character and its own quiet beauty. And there is something in that trajectory, from bare metallic ground to living community, that is deeply satisfying to contemplate as the night deepens and the mind drifts toward sleep. Things can be repaired, slowly, imperfectly, but genuinely. The world is always moving in the places where it is left to do so, toward greater complexity and life. And the small plants, with their extraordinary chemistry, are part of that movement, doing their part without fanfare, without recognition, in the quiet dark of the soil and the soft light of 1,000 different dawns. Selenium is not a metal in the strictest chemical sense. It is what chemists call a metalloid, sharing properties of both metals and non-metals. But it behaves similarly to metals in soils, and there are plants that accumulate it with the same extraordinary efficiency that other plants show for nickel or zinc. The selenium accumulators are found mainly in the western regions of North America, in areas where the bedrock geology contains elevated levels of this unusual element. They include certain species of astragalus, the milk vetches, a large genus of legumes with small, soft flowers in shades of pink, purple, and white. Some species of astragalus can accumulate selenium to concentrations high enough to make them toxic to livestock, and for a long time, they were known primarily as a threat to grazing animals. A plant that caused a mysterious condition called blind staggers, or alkali disease in cattle and sheep that ate them in quantity. The symptoms were disturbing: loss of coordination, blindness, sometimes death. Ranchers in selenium-rich areas learned to identify these plants and to manage their livestock away from pastures where they grew. The plants had a reputation as dangerous, a problem to be managed. But the same concentrations of selenium that made these plants dangerous to livestock also made them scientifically fascinating. Why were certain species accumulating selenium at all? What was the mechanism? And what was the benefit to the plant? Researchers who asked these questions found, as so often happens when you look carefully at a problem, that the answers were more complex and more interesting than the original framing suggested. Selenium, in small amounts, is actually beneficial to plants. It seems to play a role in protecting them against oxidative damage, the cellular wear and tear caused by reactive oxygen compounds that form as a byproduct of photosynthesis and other metabolic processes. Plants that have access to moderate amounts of selenium appear to handle these reactive compounds more effectively. And the defense hypothesis applies here too. Insects and pathogens that might attack selenium-rich plants find the chemistry inhospitable. The same concentration of selenium that causes problems for a grazing animal is also a deterrent to many of the small organisms that would otherwise eat the plant's leaves or invade its tissues. The flowers of selenium-accumulating astragalus species are not unusual to look at. They are typical legume flowers, somewhat like small sweet peas, delicate and soft. In spring, on the dry hillsides and plains of Wyoming, Colorado, and the Great Basin, these plants bloom alongside ordinary grasses and wild flowers, indistinguishable to the casual eye from their neighbors. A meadowlark perched on a nearby fence post does not know that some of the plants below it contain selenium at concentrations that would concern a toxicologist. The landscape looks ordinary. The extraordinary is in the chemistry, invisible, patient, always present. There is a quality of the Western American landscape that is unlike anything in Europe or Asia, a dryness and spaciousness that can feel both empty and intensely alive. The sky is enormous. The distances between things are large. Colors are clear and strong. The red of sandstone, the gray-green of sagebrush, the bright yellow of spring wildflowers, the pale gold of summer grass. In this landscape, the selenium accumulators grow quietly, doing their work under enormous skies, in a world of great space and stillness that has its own particular peace. The research on how selenium accumulators manage the chemistry of this element inside their tissues has revealed some remarkable parallels with other hyperaccumulators, and some interesting differences. Selenium can replace sulfur in certain biological molecules because the two elements behave similarly in chemical terms. This substitution, if it occurs in the wrong places, can interfere with the function of proteins and enzymes. Selenium accumulators appear to have evolved mechanisms to prevent this unwanted substitution, to ensure that selenium is directed into specific molecules where it does not cause harm, rather than being incorporated randomly into the plant's biochemistry. This kind of molecular selectivity is one of the most impressive aspects of hyperaccumulator biology. The plant is not just tolerating a flood of unusual chemistry. It is managing it, directing different compounds to different destinations with a precision that impresses researchers who study it. The cell is a very busy place, with thousands of different chemical reactions happening simultaneously, each catalyzed by specific proteins, each regulated by signals and feedback loops of extraordinary complexity. Into this busy system, the hyperaccumulator introduces enormous quantities of a potentially disruptive element and keeps everything running smoothly. It is an achievement that no human engineer has yet replicated in any artificial system. Do feel free to share this video with someone who loves the quiet side of the natural world, a friend who finds peace in learning about the hidden lives of plants, or someone who simply needs a gentle place to rest at the end of a long day. There is always room for one more in this quiet corner of the internet. Your body knows how to rest. It has been doing this all your life, settling into stillness, letting the day recede, allowing sleep to approach in its own time. You do not need to try. You only need to allow. The breath goes in, the breath goes out. The room is quiet around you. And the story of the selenium-accumulating milk vetches of the American West continues to unfold in your mind, soft and unhurried, like the sound of wind moving across dry grass on a warm afternoon. Manganese is an element that plants need in small amounts, but that becomes toxic at high concentrations. In some soils, particularly in certain tropical and subtropical regions, manganese is present at elevated levels, locked in minerals that dissolve under specific soil chemistry conditions and become available to roots. Most plants respond to high manganese availability by excluding it or by limiting its accumulation. But there are trees in the wet forests of Queensland, Australia, in the genus Gossia, that have taken the opposite approach, accumulating manganese in their leaves at concentrations that researchers have found to be among the highest recorded for any plant. These trees grow in dense subtropical rainforest, where the canopy is thick and the understory is dim and cool. Their leaves are broad and dark green, unremarkable in appearance. But when analyzed, those leaves contain manganese at concentrations that place them clearly in hyperaccumulator territory. The manganese is distributed within the leaf tissue in a way that, once again, suggests careful management, not randomly dispersed through all cell types, but concentrated in specific locations that minimize its impact on the leaf's functioning. The forests of Queensland are among the most biodiverse in Australia, and the manganese-accumulating Gossia trees are part of a community that includes hundreds of plant species, thousands of insect species, birds of extraordinary beauty, and a soil biology of immense complexity. The trees are embedded in this community. Their fallen leaves feeding the decomposer organisms of the soil, their flowers feeding pollinators, their fruits feeding birds that carry their seeds to new locations. The fact that their leaves contain unusual manganese concentrations is just one aspect of what they are, an aspect that is invisible to the birds that shelter in their branches, the fungi that colonize their roots, the insects that visit their flowers, and the rain that falls on their leaves and runs in thin streams down through the forest to the rivers below. Let the image of that wet forest fill your mind gently, the dripping of water from leaf to leaf, the soft sound of rain on a broad canopy, the dim green light filtering through layers of vegetation, the smell of earth and growing things. This is where our manganese trees live, quietly, year after year, accumulating their metal, contributing their part to the slow chemistry of the forest, asking nothing from the world except water and light and the minerals that their roots know how to find. The seasonality of hyperaccumulator plants is something that researchers have studied with interest. Metal accumulation is not constant throughout the year. It varies with the growth cycle of the plant, with soil temperature and moisture, with the metabolic activity of the roots. In some plants, metal concentrations in leaves are highest during periods of rapid growth when the transport machinery of the plant is most active. In others, Concentrations peak later in the growing season, as growth slows and metals continue to accumulate while translocation decreases. Understanding this seasonality is important for practical applications like phytomining, where the timing of harvest affects how much metal can be recovered from the crop. Spring is when many hyperaccumulators begin their most active phase of metal uptake. As temperatures rise and day length increases, the plant's metabolism accelerates, roots become more active, and the molecular pumps that pull metal ions from the soil into root cells begin working at higher rates. The metals move from roots into stems, from stems into leaves, accumulating in the vacuoles of leaf cells as the leaf expands and matures. By the time a leaf is fully developed, it has reached something close to its final metal concentration, a concentration it will maintain, more or less, until it eventually falls from the plant and returns its accumulated cargo to the soil below. The cycling of metals through plant tissue and back into soil is one of the ways that hyperaccumulators influence the chemistry of their environment over time. A plant that accumulates nickel from the soil and then drops its leaves returns nickel to the soil surface, but in a form different from what it originally absorbed, associated with organic molecules from the decomposing leaf tissue, potentially more or less available to the next generation of plants, depending on the conditions of decomposition. Over many generations, in a place where hyperaccumulators have been growing for a long time, the surface layers of the soil may develop a distinctive chemistry, richer in organically bound metals, with a different microbial community than the subsoil below. The plants are writing the history of their presence into the earth itself, slowly and invisibly, season by quiet season. Let that thought settle softly as you breathe. The earth remembers. Even when we cannot see the record, it is there. At the molecular level, the difference between a hyperaccumulator and an ordinary plant comes down, in large part, to a family of proteins called metal transporter proteins. These are molecules built from long chains of amino acids, folded into precise three-dimensional shapes that allow them to span the fatty membrane of a cell. On one side of the membrane, a metal ion, a charged atom of nickel or zinc or cobalt, approaches. The transporter recognizes it, binds it, and carries it through the membrane to the other side, where it is released into the cell's interior. It is like a molecular revolving door, purpose-built to allow specific metal ions to pass through a barrier that would otherwise stop them. In ordinary plants, these transporter proteins exist, but are expressed at low levels and regulated carefully. The cell does not want too much metal inside. Even essential trace metals like zinc and copper become toxic at high concentrations, and the cell's systems are tuned to keep them within a narrow, safe range. But in hyperaccumulators, the genes encoding certain transporter proteins are expressed at levels many times higher than in ordinary plants. The transporters are more numerous, more active, working at a higher rate, pulling more metal into the cell than ordinary cellular needs would require. This overexpression is one of the key molecular features that distinguishes hyperaccumulators from their non-accumulating relatives. How did this overexpression evolve? The details vary between species, but the general answer involves mutations in the regions of DNA that control gene expression, the promoters and other regulatory elements that determine when and how much a particular gene is active. A mutation that increased transporter expression slightly might have given a plant a small advantage in a metal-rich environment, allowing it to neutralize metal toxicity more effectively by pumping metals into vacuoles faster than they could cause damage. That advantage, repeated across many generations, would gradually be amplified until the expression levels reached what we observe in today's hyperaccumulators. This kind of regulatory evolution changes not in the protein coding part of a gene, but in the part that controls how much protein is made, is one of the most important mechanisms of adaptation in biology. It is more flexible than changes to the protein itself, because the same protein can serve different functions in different amounts. A transporter that works well at low expression levels can become the basis of a hyperaccumulation system simply by being expressed at higher levels without any change to the transporter's structure or function. The raw material was already there. Evolution just turned up the volume. The comparison to turning up a dial is actually quite apt. If you imagine all the molecular machinery of a cell as a complex mixing board, with hundreds of dials controlling the levels of different activities, then the evolution of hyperaccumulation involved turning up a specific set of dials, increasing the activity of metal transport and sequestration while leaving the rest of the cellular machinery running at normal levels. The result is a cell that functions normally in most ways, but handles metals very differently, pulling them in aggressively, moving them quickly to vacuoles, keeping them away from sensitive parts of the cellular machinery, and managing the whole process with a precision that makes the extraordinary seem routine. The genetics of hyperaccumulation are being studied in increasing detail as gene sequencing has become cheaper and faster. Researchers can now read the entire genome of a hyperaccumulator plant, all of its genetic information, and compare it in detail with the genome of a closely related non-accumulating species. These comparisons have revealed the specific genes that are expressed differently in hyperaccumulators and have begun to identify the regulatory changes that underlie those expression differences. This knowledge is valuable for understanding the biology of these plants, but it is also valuable for practical applications. If you know which genes are responsible for hyperaccumulation, you can begin to think about whether those genes could be introduced into other plants, or whether the plants themselves could be modified to accumulate more metal, or to grow faster, or to be better adapted to the specific conditions of a contaminated site. The idea of using genetic knowledge to improve plants for phytoremediation is an active area of research. Scientists have experimented with introducing metal transporter genes from hyperaccumulators into faster-growing, larger biomass plants like tobacco or mustard. Plants that might accumulate metal less efficiently but produce so much more tissue that the total amount of metal removed per hectare could be greater. The results of these experiments have been mixed, and the regulatory and public perception challenges around genetically modified organisms add complexity to any practical application. But the research continues, driven by the recognition that in the genes of hyperaccumulators there is knowledge encoded over millions of years that might be genuinely useful if it can be understood and carefully applied. Please do visit our playlist if you are looking for more evenings like this one. There are many quiet journeys waiting there through forests and oceans and the hidden lives of animals and plants. Each one is made with the same care and the same hope that it will find you at the right moment, when the day has been long and the need for stillness is real. The night is deep now, or perhaps the evening is soft and early, or perhaps it is the middle of a quiet afternoon and you are resting. Whatever the time, whatever the light outside, the story goes on at the same unhurried pace. The breath moves in and out. The mind drifts, a little looser now than when we began. The body is heavy and warm. These are the conditions in which the best kind of learning happens. Not effortful or pressured, but received, like warmth from a fire, like the smell of rain, like the sound of a distant river that you hear just as you are falling asleep and carry with you into the dream. Nickel is the element that has inspired the most research on hyperaccumulation, partly because it is economically valuable, and partly because there are more known nickel hyperaccumulators than hyperaccumulators of any other single element. Researchers have cataloged hundreds of nickel hyperaccumulating species, from small herbs in the Mediterranean region to large shrubs and trees in the tropics. They are found on ultramafic soils derived from rocks like serpentinite and peridotite that are naturally rich in nickel, magnesium, and other elements, on every continent except Antarctica. The global distribution of ultramafic soils is itself a fascinating reflection of the Earth's geological history. These soils occur where pieces of the oceanic crust or upper mantle have been thrust above sea level by the forces of plate tectonics, a process called abduction, or where deep volcanic activity has brought mantle material close to the surface. The result is a scattered geography of unusual soils, disconnected from each other by vast areas of ordinary geology, each patch an island of unusual chemistry in a sea of more typical Earth. And on each of these geological islands, a community of specially adapted plants has developed, often with high levels of endemism, species found in that location and nowhere else in the world. This pattern of unusual plants on unusual soils, each community isolated from others by the distances of ordinary geography, means that the hyperaccumulator floras of different regions often have no direct evolutionary connection to each other. The nickel hyperaccumulators of New Caledonia are not closely related to the nickel hyperaccumulators of the Philippines, which are not closely related to those of Cuba or Greece or South Africa. Each group evolved independently from different ancestral species in response to the same chemical challenge. The result is a kind of global experiment in convergent evolution. Dozens of separate tests of the same question, each arriving at a similar answer through a different path, and scattered across the earth. In all these separate places, the plants grow quietly, carrying their nickel, living their slow and patient lives, unaware of each other, unaware of us, simply present in the world that made them. The island of Cuba is one of the places with a particularly rich hyperaccumulator flora. Its geology includes extensive areas of ultramafic rock, and the flora that has developed on the associated soils over millennia is extraordinarily diverse and highly endemic. Researchers working in Cuba have documented numerous hyperaccumulating species, many of them known only from this island, found growing on the reddish soils of its ultramafic regions in communities of great botanical richness. The Cuban hyperaccumulator flora is considered one of the most species-rich in the world, and it is increasingly a focus of research both for its biological interest and for its potential applications. To walk through a Cuban serpentine barren is to walk through a living library, a collection of plants, many found nowhere else on Earth, each one the product of a particular evolutionary history in a particular place. The landscape itself might look sparse to a casual observer, open, rocky, with scattered shrubs and herbs rather than the dense vegetation of more fertile soils. But to a botanist, it is extraordinarily rich. Every plant a story, every species an experiment in adaptation, every root system a record of the long conversation between life and the unusual earth of this place. Breathe in slowly. Breathe out. Let the image of that open landscape, the reddish soil, the scattered plants, the enormous sky of a Caribbean day, drift through your mind like a warm breeze. You are here. You are quiet. The story continues, unhurried, just ahead. The sunlight in the Mediterranean falls differently than it does elsewhere. It has a particular quality, bright and direct, without the haze that softens light in more humid climates, falling on limestone and scrubby vegetation and the silver-gray leaves of olive trees. In parts of Greece, Albania, and Turkey, where the bedrock contains serpentinite and other ultramafic rocks, this Mediterranean light falls on plants that are quietly managing some of the highest nickel concentrations found in European soils. Among these plants are species of alyssum, small, mat-forming plants with clusters of tiny yellow flowers that bloom in spring and carpet certain rocky hillsides with color. Alyssum is a large genus, and most of its members are ordinary enough. Cheerful little plants, tough and drought-resistant, growing on rocky ground around the Mediterranean. But certain species within the genus, Alyssum bertoloni in Italy, Alyssum murale across the Balkans and Turkey, are nickel hyperaccumulators of the first order, capable of accumulating nickel to levels that clearly qualify them for the most extreme category of hyperaccumulation. Alyssum murale in particular has been studied extensively for its potential use in phytomining, because it grows readily, produces a reasonable amount of biomass, and can accumulate nickel at concentrations that make the economics of phytomining calculations look interesting. Researchers in Oregon and Nevada have conducted field trials of Alyssum murale as a phytomining crop on nickel-rich ultramafic soils. The plants were grown across several growing seasons, harvested, and the metal-rich biomass was analyzed. The results suggested that under appropriate conditions, yields of nickel from alyssum cultivation could approach those that would make phytomining economically viable. Not everywhere, not in all conditions, but in specific scenarios where the soil nickel content is high enough and the other conditions of plant growth are met. These trials were a significant step in moving phytomining from a theoretical concept to something that might actually be deployed. The biomass of a metal-rich crop like Alyssum murale is sometimes described as a phytoore, a biological ore analogous to a metal ore in the sense that it contains metal at concentrations worth extracting, but in plant tissue rather than in rock. Processing a phytoore is different from processing a conventional ore. You cannot use the same high-temperature smelting techniques because burning metal-rich plant material releases the metals as volatile compounds that are themselves potentially hazardous. Instead, researchers have experimented with approaches like using acid to leach metals from the ash of burned plant material, or with biochemical extraction methods that dissolve metals from the organic matrix of dried and ground plant tissue. The chemistry is complex, and finding cost-effective and environmentally safe processing methods is one of the key challenges facing phytomining as a practical technology. But these are engineering problems, and engineering problems have a way of being solved when the motivation is sufficient and the underlying biology is sound. The biology of Alyssum murale is sound. It is well understood, well documented, and genuinely impressive in what it can accomplish in terms of metal accumulation. The engineering will follow in time, driven by a combination of economic opportunity and environmental necessity. The spring flowering of Alyssum murale is one of those spectacles that rewards the observer who happens to be in the right place at the right time. A hillside covered with these small plants in full bloom is covered with yellow, not the deep yellow of sunflowers or the pale yellow of primroses, but the bright, clear, warm yellow of a plant that has caught the Mediterranean spring at its fullest. Bees visit the flowers eagerly. The air is warm and smells of sun-heated stone and the sweet, faint fragrance of small flowers. And beneath all this color and fragrance, in the soil and in the roots and in the leaves of these blooming plants, the quiet chemistry of nickel accumulation continues without pause. Invisible, patient, perfectly adapted to its work. You might feel right now a kind of softening in your muscles, in your thoughts, in the quality of your attention. This is the natural direction of rest. The body knows how to get there, and the mind follows, especially when the evening is quiet and the voice is soft, and the story is about things that are slow and patient and deeply at home in the world. You are moving in the right direction. Let it happen without effort. The Mediterranean hyperaccumulator flora extends beyond Alyssum. In parts of Italy, there are populations of Noccaya caerulescens on serpentine outcroppings, the same small plant we met in the Belgian meadows, but here growing in a warmer and drier environment. Adapting as hyperaccumulators have always adapted to the specific conditions of their place. In Greece, researchers have documented species of thlaspi, another genus in the mustard family, growing on nickel-rich soils and accumulating nickel to hyperaccumulator levels. In Turkey, new hyperaccumulating species are still being discovered as researchers explore the botanical diversity of that country's complex geology. Turkey is particularly interesting from the perspective of hyperaccumulator research because it sits at the intersection of several tectonic plates and has a geology of remarkable diversity. Its ultramafic soils are widespread in certain regions, and its flora, shaped by its position as a bridge between European and Asian botanical regions, is extraordinarily rich. Estimates suggest that Turkey has more plant species than the whole of Europe combined. And among those species, researchers continue to find new examples of metal-tolerant and metal-accumulating plants. The discovery of a new hyperaccumulator is not a dramatic event. It begins with a researcher noticing a plant growing where it perhaps should not, in poor soil, in a place where the chemistry of the ground should be hostile to most vegetation. The plant is collected and dried, and samples are sent to a laboratory for analysis. Weeks later, the results come back. The metal concentration in the leaves is 10, 50, 100 times higher than normal. A few more samples are collected from more plants, in more locations, to confirm that this is real and reproducible. A paper is written, peer-reviewed, and eventually published. The new hyperaccumulator takes its place in the scientific literature. One more entry in the growing catalog of plants that have found a way to make something extraordinary out of the Earth's difficult chemistry. And somewhere, at the moment, a researcher reads those analytical results for the first time and realizes what they are seeing. There is a moment of pure, unadorned wonder. The same wonder that is present every time the natural world reveals something unexpected. Something that reminds us how much more there is to know. How many quiet marvels are present in the world if we are patient enough and curious enough to look for them. That wonder does not have to be loud or urgent. It can be just what it is, a gentle expansion of the sense of what is possible, a small brightening of the awareness that the world is richer than any list of what we know about it.